Dr. Zavos may be confident, but the more the scientists examined different organs from dead clones, the worse the picture became. They found a bizarre array of deformities never seen in nature. On the left is a cross-section of a kidney from a normal lamb. On the right, on the same scale, are cross-sections from two clones. These kidneys are shrunken, with much of their tissue missing. And there were other problems. A normal lamb's liver is mostly made up of healthy liver cells, seen here in pink. But in the clones, most of the liver was made of strange, unspecialized cells that couldn't function as liver cells. The implications for human cloning were terrifying. If a human baby born with these kinds of defects were to survive at all, many scientists now believe it would suffer devastating illnesses unknown to medicine. But Dr. Zavos and his Italian associate, Dr. Severino Antinori, are confident they can produce healthy human clones.
We have no intentions to step over dead bodies or deformed babies to accomplish this.
I am not sure to permit to continue the pregnancy if there is some abnormality.
And the prospect of having an abnormal child is not enough to deter people desperate for a family. Couples like Desiree and Matthew seem to have no idea how profound the problems might be.
But even if it was something wrong with the child, we wouldn't toss the child away. We'd both still raise it to the best of our ability and treat it as any normal child.
That would be like me if I could get pregnant myself and we found out at 5 months that it didn't have an arm or a leg or something, and then telling us we could abort it. I don't really think we would. I don't see us being that type of people. We created it. Sure. We're gonna, it's our responsibility.
But the scientists who had seen the scale of the problems knew they had to get to the bottom of what was going wrong. They tested many theories. Perhaps clones made from adult cells were just dying from premature old age. Perhaps cloning was causing strange mutations in their genes. But everything they looked at seemed normal. Many began to suspect that the problem lay in the great mystery at the heart of cloning. How could a single cell taken from an adult make a whole new animal, or human?
We have a system which has evolved over millions of years to make fertilization work very effectively, usually. And what we're asking it to do is something totally different. So really we shouldn't be surprised that it doesn't work very well. In a way, almost we should be surprised that it does work sometimes.
The way we are made is incredibly complex. Our genes are like the plates on a printing press. Each gene is a template that produces a message. To operate, they need to be switched on. And when they're switched on, the different genes produce different messages that tell the cells how to build all the organs and tissues we need. To make a healthy human, our genes need to be switched on at the right time, in an intricate cascade. And crucially, when a process is complete, they need to be switched off again. But the bizarre defects in cloned animals, like the massive blood vessels, suggested that something was going wrong with these critical instructions. So researchers began the quest to discover the problem. The first clue was to come from a scientist who wasn't doing cloning, but was working in a related area, IVF. For years, scientists had been puzzled by the fact that some animals made by IVF for selective breeding grew dangerously large in the womb, something that also happens to clones. When Lorraine Young first set out to discover what was going wrong, it seemed an impossible task.
To try and pinpoint what was going wrong, it was essentially a needle in a haystack type exercise. There are about 30,000 genes in every cell in an embryo, and really any one of those could be going wrong.
The answer to why the animals were growing too big could lie anywhere among 30,000 genes. But Dr. Young knew of something which might help narrow the search. It was a rare disease in humans, Beckwith-Wiedemann syndrome. Thelma was born with symptoms of Beckwith-Wiedemann syndrome. It's a disease in which a vital few genes that control how we grow go wrong.
Bye-bye. Bye-bye. When Thelma was born, she ended up being born with a part of her intestines formed outside of her stomach wall, and she required surgery within 1 or 2 days of having been born.
Beckwith-Wiedemann syndrome causes dangerous errors in the way babies grow. They're often born very large, with huge tongues, and other problems that have to be corrected with surgery. And cells in the liver and kidney can also grow out of control, causing tumours. All this is caused by an error in the functioning of one of a small number of genes that control how babies grow. As a result, too much growth factor may be produced, which in turn causes the tumours and abnormal growth.
Good job.
Thelma will have to have ultrasound scans every 3 months until she is 8 to check for tumours.
Well, we try to go into these screenings very optimistic every time, but you never know if it's going to be okay until the doctor, takes his look and says it's okay. And you try not to think about what it would be like if they came back and said it's not okay, but it means cancer. You don't want your child to go through that.
Now, Lorraine Young had a hunch that the same group of genes that cause problems in children like Thelma could also be making the IVF animals and clones grow too big. She began comparing the chemical signals coming from these genes in healthy lambs and overgrown lambs. It would take many months of painstaking work. But finally, she found a crucial difference. One of the genes she was studying is meant to keep growth under control. But Dr. Young found it wasn't producing strong enough signals. And when she analysed it, she found that something vital was missing. The switch that should turn the gene on, a tiny molecule called a methyl molecule, wasn't where it should be. Without this switch, the gene wouldn't work, and the animal would grow out of control.
This observation shows that loss of these methyl groups from the DNA was enough to make the animals grow large, and this was our most important result to date.
Dr. Young had done her work in IVF animals, but perhaps the same thing was going wrong in clones. Perhaps the abnormalities were caused by switches like methyl molecules being disturbed. To test this theory, scientists around the world began to look at tissue from clones to see whether methyl molecules had gone awry.